How many of you have had statistics? How many of you not had statistics? How many of you are not voting? Okay. All right. Well, actually, that's good. We're going to talk a little bit about statistics today. I'll give you a little pre-lecture. What the focus of this, one of the center points of this lecture is going to be about a technique that's called regression. And this was invented by this guy, Sir Francis Galton. And Galton was Darwin's cousin. I recently found out that one of Sir Francis Crick, remember Watson and Crick? One of his ancestors was Darwin. Seems to run in these families. At any rate, he came up with this statistical technique. And one of the things that he discovered in this statistical technique, and the reason why it got its name, is he noticed that everyone, or that through time, populations, or individuals rather, the offspring of individuals, regress towards the mean. And this is a phenomenon that we find, and that's because out at the outliers in, say, a population, someone who's fortunate enough to be born very short, well, that's because they got the right shuffle of very short genes, okay? And it's unlikely that in the next generation you're going to get that same kind of shuffle to make someone very short or correspondingly very tall. And similarly, this also works with sports teams. So it's part of winning Super Bowls is a little bit of luck. And so no one's going to get that lucky time after time after time, year after year after year. In fact, I can tell you way back when the Green Bay Packers used to be the team that dominated football, and now they're not doing very well, right? Right. So everyone kind of shuffles in towards the mean through time. So part of it's luck, just being out there on the outliers of luck. And Galton discovered this, and I don't know how he came to reconcile this with his what are called eugenics leanings. And anybody here ever hear of eugenics? No. Well, this idea caught on, especially in this country. And the idea was, well, we're breeding cattle and dogs and sheep for special characteristics. Maybe we ought to think about doing this for people too. And so maybe there are some people we don't want to breed because, say, they're mentally defective. So let's try and make sure they don't breed. So some people in this country got sterilized without their permission so they wouldn't breed. This idea, by the way, is back. It's dressed up in new clothes. But anyway, I'll leave it right there. So that's my pre-lecture, and now we'll get to the regular lecture. All right. In your reader, there are a couple articles. This one by Judith Grisel, who I had the pleasure of meeting, is a really good article on quantitative trait locus analysis, very readable and very thorough. She does a great job of it. So please give this a look, and this should make more sense to you if you do. And what we are trying to do in this lab, also in your reader, you'll find this paper by Williams et al. And what we are trying to do in this lab is replicate their results, okay? So you should probably at least give this a peek. You don't necessarily need to know every detail in this, but you should kind of have some sense of what they got, all right, by way of results, because we're going to compare our results to theirs and see if we come up with similar answers. Okay, to get you up to steam, this module, actually I should probably retitle it the Bioinformatics module, but what we did so far is we quantified the phenotype, we measured the olfactory bulbs, and now what we're going to do is we have set to already getting rid of some error variance, and I'm going to talk later about why error variance is so deadly to this kind of analysis. So one of the ways, one of the problems that we had was differential shrinkage of the brain. So they shrunk them down to section them, and the brain shrank differentially. And we also had multiple raters. So everybody had somebody else who was doing at least one other person, sometimes two other people, who were measuring the same set of brains. So that can actually also, even though it's a check, it also can create error variance. So here my buddy Gregor wants to get up and clean up these data. He wants to know who shrank the brains, and most of all, what do we do about it? Okay, so what do we do about shrinking the differential shrinkage? We ran it through this equation to get everybody back to their original state, right? Okay, so that's how we got past that hurdle. And also now, how did we get past the hurdle of the fact that we had multiple raters? What did you do to get rid of the error variance that this would introduce? What did you do last Thursday? You have no idea what you did last Thursday. All right, for those of some of you got to go home because you did a great job. For those of you who didn't get to go home, you had to sit down as a jury and hammer it out and figure out why you were not within 10% of the mean of each other. Okay? So we made you figure it out. Part of this was my experience when I was an undergraduate. I may have mentioned my lab partner, Dougie, who always had hemp flakes for breakfast. And Dougie wasn't really always on target, and so he got the weirdest data you can imagine. So if we had somebody like that, hope we didn't, we could take care of that. All right, so as my buddy Gregor points out, we insisted that these raters agree. You had to get within 10% of the mean. And what we're also going to do is even so, we're going to use the median value of all measures for a given mouse. Now, in most cases, we had 2 people measuring a given mouse, so the mean and the median happened to be the same exact number. But in one case, we had 3, so we use the median. Why would we choose the median rather than the mean? For those of you who had statistics, go for it. By outliers, right. So if you had somebody like my partner Dougie, whose data was way the heck out in the boondocks, you use a mean, those data that are way out the mean is influenced by those data. Median, no, it's not. So we use the median. And in one case, we did have 3 people rather than 2. So most of the time, it doesn't matter. Mean, median, if you have 2, it's the same. But 3, we're going to use the median. So that's how we're getting rid of the error due to multiple raters, at least some of it. All right, now, we also are going to have a problem, as my buddy Gregor is going to point out. We've got a lot of extraneous variables, what I'm calling demographic characteristics. Things like sex, age, body weight, and indeed brain weight are different among these mice. So let's see what he has to say about it. Okay. Once we get that cleaned up, by the way, we're going to go on and link the variants to chromosomal markers, determine genes, and have you find a gene that's highly regulated and then go in depth. But at any rate, my friend Gregor says, all right, you quantified the phenotype, let's get on with it, and then I showed him the data. So here's some data. This actually is just a piece of the spreadsheet, and what you can see in this spreadsheet is here we've got all of our different strains. By the way, we are using some F0s, so there are some Ds and Bs, okay? But all the rest of these BXD24, these are all recombinant inbred strains. And you can see we've got both males and females. Well, could that possibly matter with regard to how big their olfactory bulbs are? Yeah. And you can see we have a huge range of ages. That 92, that's a pretty mature mouse. We have this little boy here who's only 21 days old. He hasn't gone through puberty yet. Okay, so we got a huge range in ages. Could that possibly influence the size of olfactory bulbs? You bet. And accordingly, we got a huge set of body weights, 21 and some grams. Here, this little girl's only 14 some grams, 13 grams for this little girl. Huge range, all right? Could that have an influence on olfactory bulbs? Yeah. And also, something that Williams et al. Did was they also controlled for brain weight. And their rationale was they wanted to make sure that what they were looking at was specific to olfactory bulbs and not just brains as a whole, all right? So actually, I'm not sure that was a great idea, but we're going to do it because we're going to try to replicate their experiment, all right? So my buddy Gregor says, you got a mess here, all right? Why did you use mice of all different sexes, ages, sizes? Why would you ever do that? Didn't your professors ever teach you anything about controls? All right, so let's talk about that. If you have a good control, what is different between the manipulated group and the control group? Say it loud. One variable, right? You don't let all this stuff vary, okay? Why? Couple of reasons. One, attributions of causality, right? If you only have one thing different, then you can say, okay, we got a causal relationship here for the difference. And two, if you let all this stuff vary all over the place, then you have a bigger chance of not getting a significant effect, okay, because you have all these variable scores. You have a bigger chance of not finding any positive results. So, what that means is we're going to want to try to control for all that. And this is where Francis Galton comes in, Chuck Darwin's cousin. And so what he invented was this tool called linear regression. I didn't tell you in my little pre-lecture. What he used to do was he'd go around all these county fairs, and people would actually pay him to get themselves measured in some way, their arms or their head size or all kinds of things. They'd pay him to get data, and then he would tell them how they matched up with the whole population. So he got huge amounts of data. All right. One of the things, he was actually a really smart guy. He came up with linear regression, and what we're going to do with this tool, linear regression, is we are going to use this to control for these extraneous variables. Okay, so obviously at this point, we can't control for sex. You got males and females, can't control for body weight and brain weight and age, but what we're going to do is we are going to control for these things by means of the statistical method, linear regression. We can do this after the fact, okay? I'm going to pause here and step back and show you that this actually has applications to real life kinds of research, because if you're doing research on clinical populations, you get them as you get them, both male and female, all kinds of ages, all kinds of sizes, et cetera, and you might want to control for all this, and you can't say you randomly assign people, then perfectly match them. So one thing you can do is this control by linear regression, or as it's going to be for us, multiple regression, and take the influence of those variables out. That's what we're going to try to do. It's not a perfect way to do it, by the way. There are some big drawbacks that we'll talk about, but it is a way to do it. So what we're going to do is use linear regression to eliminate the variance. And again, I'm going to ask one more time. How many of you have not had statistics? Excellent. Okay, so I can say things like variance, and nobody's going to freak out, right? So variance, we can get rid of the variance that's associated with these extraneous variables that we don't care about, that could be confounding our experiment. All right, so the big assumption that he made is that the variance, which is indeed statistics talk for individual differences, is additive. And with that little bit of assumption, it's going to be very powerful for us. All right. So what we come up with is what's called this additive model of variance. So if I use, you told me you all had statistics, so if I make the sigma squared, you all understand that means variance. Yep? Excellent. All right. So if we look at what Galton came up with is you can partition variance. You can break it up into components. And so our total variance, we could break it up into components. Some of the variance is due to sex. Some of the variance is due to body weight. Some of the variance is due to brain weight. Some of the variance is due to age. Some of the variance is unique to olfactory bulbs. And of course, the bad news is some of the variance is always due to error. All right? So what we're going to try to do is use this technique to get rid of the variance that's due to sex, to get rid of the variance that's due to body weight, to get rid of the variance that's due to brain weight, to get rid of the variance due to age, and that's going to leave us with the variance that's unique to olfactory bulbs, and unfortunately, error 2. All right, so that's the plan. Okay, so as I just said, you can partition the total variance associated with each of the variables, and then what we're going to do is we're going to successively remove the variance associated with all these variables. And then what we'll have at the end is something called the residual variance. The residual variance, and that's what's left over, and that will have the variance that's unique to olfactory bulbs, that residual variance, and of course there's going to be a little bit of error in there too, hopefully a little bit. We'll find out. All right. Before I get into that, so what we're going to do, by the way, when we take these things out all at once is something called multiple regression, which I suspect most of you didn't study. Most intro stats class don't get into that, but it's just kind of a special case of simple linear regression. So let's talk about that first and remind you of something you probably already know. So we're going to talk first of all about simple linear regression where we only have two variables, a predictor variable and a variable we're trying to predict. Okay. So what we are doing in regression is we're trying to predict the y variable from the x variable. Okay? And what I've put up here is a whole bunch of different kinds of scatter plots. And what you can see, the data are all these white points, and you can see that through these points, each of these plots has a line, and that's the regression line that's drawn there. So there's another statistic that goes along with regression, and we'll talk about that also in this unit, and that's correlation. Okay? And I presume you all studied correlation oh, great. Okay, this is going to be so easy. All right, so if all of the points are on the regression line, the correlation is what? 1. And how much of the variance is accounted for? 100%. Right. Okay. Trick question. It was too easy, right? Okay. So as our predictors get worse and worse, of course, the points are going to get further and further and further off this regression line. And as they get really off this regression line, the correlation goes to what? 0. And the way I was taught, if you can draw a 0 around your scatter plot, that's pretty much what your correlation is. Okay? So that's a correlation of minus 0.10. That's a weak relationship. Still could be significant, by the way, but that's another lecture at another time. I got a couple of squirrel cases here, unfortunately. If you could have all the points on the line, but you can see here that your y variable doesn't predict any different, your x variable rather, doesn't predict any difference in your y variable, all right? And so there your relationship is still a 0, all right? And this is a real squirrel case, but one that reminds me to say that regression and correlation are talk about the best linear relationship between two variables. Here we have a very obvious curvilinear relationship, but it says it's a linear relationship is zip, all right? Okay, that's kind of a squirrel case. You don't really see this very often. At least I don't. Okay, so let's now talk about this regression line. If we do have a whole bunch of points like this in a scatter plot here, they're trying to predict weight from height. You could think about, you could draw all kinds of different lines through there, but what we would want to draw through there is a line that best characterizes the relationship between height and weight, trying to predict weight from height. So what we're going to do is we are going to find a line that is called the regression line. It's also called the least squares line, also called the best fit line. And what this line does is, of all the possible lines we could possibly draw through this particular plot, that is the line that minimizes the square deviations in the y variable. It's the one that minimizes the square deviations of the y variable from that line. Okay? So the y variable then it's the one that best predicts the y variable of all the possible lines. Does that make sense to everybody? Okay. So it's the one that minimizes the square deviations in the y variable. So that's the regression line, or the least squares line. Okay, I put this slide up to point out that usually what one uses regression for is to predict one variable from another. Okay, so someone sat down and tried to predict what's GPA going to be at UCLA given SAT scores. Okay, something like that. And to the degree that they miss, that's the residual. And usually people think of the residual as the slop when you miss. All right, but for us, the residual is gold, because that's where the variance due to the olfactory bulbs is going to be. So usually when you use this statistic, the residual is the junk, but for us, it's treasure. All right? Okay, so let's look at kind of a concrete example so you can kind of wrap your head around what we're doing here with this. And again, I've just got out two variables. This is simple linear regression, but multiple regression would work much the same way. So what we're trying to do is predict olfactory bulb volume from our predictor variable. In this case, we'll take body weight. All right? Now, just let's pretend. What if body weight did not predict olfactory bulb volume at all? It does, but let's pretend for a moment it doesn't. Okay? What would be the best prediction then of the value of Y? If you've got to reach in and grab an olfactory bulb and weigh it, or measure its volume rather, what would you predict? You got to make a bet. What are you going to bet on? Okay, why did you pick 18? Centered, and not only that, but it is the mean. Okay, so if your predictor variable doesn't predict, then the best guess is the mean of Y. Make sense? All right. What if? What if your predictor variable does predict something about the Y variable? You can do better than the mean of Y. Okay? So what I've done to illustrate this is to show you that we're going to do better than the mean of Y. And so here, we're going to take this one point here, happy face point, which I selected out of all the points in this. And so for this particular mouse that weighed almost, say, 37 or so grams, we would predict its olfactory bulb volume to be, what, say, about 24 or so, okay? So we do better, right, by having this predictor variable. And so we would predict that much, the whole distance of this point from the mean if we squared it, would be the total variance for that point, right? Yep. Say yeah. So we predicted that much of the variance. Check? Okay. And so we missed by a little bit. We got a little residual on this point, some error. Well, I'm just going to try and call it residual. So what we can do, though, is we can say, all right, we don't want the variance due to body weight to influence our scores. All right? So we're going to go through and do this for every last point. Figure out the variance that's predicted by body weight and throw that out. So we're going to control for body weight. So we're going to make it like everybody had the same body weight. Does that make sense? So what we're going to do here is throw out the variance. We're going to figure out what the variance due to body weight is for every last point in this distribution. Throw that variability out. Throw that variance out. Okay, and so control for body weight. Does that make sense? Okay, so that's it. That's essentially how we're going to do this, except for we're going to do it for 4 variables at once, rather than just body weight. Okay. So what we'll end up with is the residual variance, the variance left over after the variance of the other variables, or variables in our case, has been removed. And again, it's precious to us because that's going to have the variance that's specific to the olfactory bulbs after we get rid of all those other things. So the variant, we're going to take out the variance that's, let's go back here a few slides. We're going to take out the variance due to sex and body weight and brain weight and age, all in one great swoop, just like I showed you, with simple linear regression. We're just going to do something called multiple regression, does the same darn thing. All right, and then we'll have the residual, which will just be the variance due to error and the variance due to olfactory bulbs. Okay? So I'm going to take a little sidebar here while this soaks in and talk about why this is not necessarily a wonderful, wonderful thing. I mean, you might think, why don't we do every experiment like this if it's that darn easy? You know, why go through, constructing all these careful controls and everything else? All right, here's the problem. And so this assumes that all the different variance is independent, right? Okay? What if it's not? All right, so let's say, what if, say, brain weight and olfactory bulb size is correlated? And you haul out the variance that's due to brain weight. What are you going to do with the variance that's due to olfactory bulbs? That goes too. All right? So that's the problem. You can be throwing out the variance that you're really interested in if they're not really independent. And here's the bad news for us. Some of our variables, I can tell you, will not be independent. Okay, but we're trying to replicate what Williams and al. did, and so we're going to do it exactly like they did, even though I think some of their presumptions are a little dubious. Okay, so that's the problem, and that is a big problem for using this as a control. But again, a lot of times, say in clinical stuff, you got no choice. This is the best game in town. All right? Okay, to continue. Okay, so for us, this is the term that represents the residual, and that's the treasure, okay, because that's got the variance that's specific to olfactory bulbs. Usually, again, when you're doing the statistic, that's the trash, okay? So we're going to remove the variance associated with those extraneous variables that we don't care about, and so statistically control for those variables because we didn't do it up front. Actually, in all fairness to the people who built the mouse brain library, they did that on purpose. So you could look at things like age or like sex or all these different variables. That's why they have everything, all these mice all over the place. So it allows for lots of different studies. They did that on purpose. But for us, it's going to create a control problem. So again, we're going to use this tool to control for sex, body weight, brain weight, and age, and get rid of the variance due to that, and hopefully distill out the variance that's due to olfactory bulbs and error. So one of the things I'm going to have you do, and by the way, I should, at the end of this lecture, talk about when the next office hours and assignment is due. One of the things I'm going to have you do is plot the variance in scores or the residuals as a function of, we're going to take all these things out kind of successively. And what you can see in this case, this was from another term, so this isn't your data. Don't copy this graph. Your data didn't quite come out like this. Somebody ran it. But you can see, in this case, sex didn't matter much, right? Okay, so the variance stayed about the same. But when we put age in with sex, boom, some of the variability, so age mattered. It accounted for some of the variance in olfactory bulbs. And here you could actually see that when we added body weight to all this, the variance actually went up a little bit. That's because of that problem of not being independent. Can sometimes blow a little variance back into your data, okay? But when we took sex, age, body weight, and brain weight out, all four at once, you can see the variability in scores went way down. So what that suggests is we are cleaning up the data and taking out the variability due to these variables that we don't care about. All right? And that's what we're up to. Okay, so That's how we're going to get rid of the extraneous variables, the demographic characteristics, we're going to of sex, age, body weight, and brain weight. We're going to use multiple regression to control for these variables. Now, we still have some individual differences, and we have several individual mice representing each recombinant inbred strain or F0 strain. Okay? And so how would we dampen down these individual differences? What could we do to get rid of the variability due to those? So we have 3 or 4 mice for each recombinant inbred strain. How could we end up with one, a summary score that would get rid of some of that variability? Just take the mean. Good answer. All right, so what we're going to do that is we are going to average across the values from the various individuals within a given recombinant inbred strain. So we'll get rid of more variance due to individual differences by doing that. Okay? Okay. All right, so that's how we're going to get rid of other individual differences. We'll just, so, you can still have individual differences, because how do these come about? Well, you have, anybody ever been in a vivarium and seen mice? Nobody? Oh. Okay, so does everybody have the same cage and the same lighting condition on the same place in the rack? No. Sometimes, some of them live on the top of the rack, some of them live on the bottom of the rack. We don't think this has anything to do with their olfactory bulbs, but we don't know what we don't know. All right, so these individual differences can arise. They do have, we try to give them the most uniform little lives that we possibly can. They all eat the same food, drink the same water, breathe the same air, but still, we can't make it perfect. All right, so now we've cleaned up the data, and now we can relate the residual variance, which we hope is specific to olfactory bulbs, to each individual marker on each chromosome except Y. In other words, do a QTL analysis, right? Oh, geez. And so that actually we would be ready at that point, but we have some other things to think about in this lecture before we leap into that. And Sir Francis comes back to remind me to tell you that each measurement has actually 2 components, and just like I mentioned, we're going to have some that are true score, which would be the variability due to olfactory bulbs, but some of that score is going to be due to error. All right? Can't get rid of it. Okay, so let's talk about what happens when we have error. And to do that, I am going to need to talk about how to make mistakes with statistics. And why do we care about this? If we have a lot of error variance in our data, how will that affect our outcome? Okay? So error variance we can think of as being kind of just adding random scores to our data, adding or subtracting random scores to our data. Okay? So I want you to keep in mind and think about what the heck is that going to do to our outcomes if we're adding and subtracting random scores to our data. Okay? So what that's going to mean is if we're trying to get some relationship between our phenotype and anything, if you got a lot of variance, what's the relationship going to be? Nothing. Even though in instances it should probably be true. All right? Possibly, hopefully. All right, so randomness can't be systematically related to anything, right? So if most of our data is randomness, we just wasted a whole lot of time. All right. So let's talk about how to make mistakes with statistics. Mistakes in statistics basically come in 2 big flavors. And let's talk first of all, what we've been talking about are type 2, which are also known as beta errors. And statisticians never use this term, but they probably should. These are essentially false negatives. This is when you say there's no relationship or no effect but there really, in reality, you should have said that there was. Okay, so these are type 2 errors. So false negatives. Okay? So, how do you get false negatives? Of course, you don't want them, but what contributes to them is, one, if you have a small effect size, okay? Small differences. For us, that means if there are small influences of a given gene on our phenotype, we may miss it, okay? Because it's just not big enough for us to pick up, all right? If you're designing an experiment, don't design something to look for subtle effects, because you could miss it just because of the small effect size, all right? As one of my mentors taught me, if you're going to design an experiment, hit it with a sledgehammer. Make sure there's some, you have big effects, then you can detect them. Don't go looking for the subtle. It's too hard to find. Okay, small n, what's that mean? Small sample size. That will be something that will contribute towards making these type 2 errors. The smaller the sample size, the more likely it is you'll make a type 2 error, get a false negative. Okay, and what I've been hammering on is greater variance in scores. The greater the error variance, the more the type 2 errors. So, in fact, just the greater the variance period, the more the type 2 errors. And you can contribute to that by making a lot of error variance, having a lot of error variance participating. Okay, hang on to that idea. All right? The other kind of mistake that one can make in statistics, the other big flavor, are called type 1 or alpha errors. These are false positives. Okay, and so these are false positives. So this is when you say there's an effect or you say there's a relationship and there really wasn't one. Okay, so for us, this is if you would say there's a peak on our QTL and there really wasn't one. Okay, so that would be a false positive. All right, or if you say there's a difference or some relationship and there really wasn't one, that's a type 1 error. Now, let me go to the next slide and talk about this. Is actually a problem in this kind of analysis. Okay. Because, and the reason why it's a problem is we are going to actually not you, but the computer is going to be doing these statistical tests over and 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 over again many times, okay? And each time it does that, there is some probability of making that type 1 error, all right? And that probability compounds every time it does it. So here you can see this is going to be the likelihood ratio statistic. That's what we're going to be calculating as a function of all these different markers. And this, by the way, is not even close to an exhaustive list. All right, there are thousands of these markers, and the computer is going to be making thousands of these tests. Okay, so that actually means that so what's going to do is it's going to go to the first marker, separate the B mice from the D mice, do the test. Go on to the next marker, separate the B's from the D's, do the test, over and over and over and over again. So every time it does it, the probability of making that type 1 error is going to compound. All right? That's a big problem because that means the false positive rate's going to do what? Go way up. All right, so we got to make sure we take care of that. So the way they take care of that is for any given individual point, every comparison, they make it extremely stringent. It's really hard to get that to the significance level. And I actually figured this out a couple years ago. Now, since then, they've added a lot more markers since I figured this out. So last time I figured this out, it was probability of 0.00006 for a given point. All right? And if you added all those up then across the whole genome, all the chromosomes, you'd end up with a probability of 0.05 across the whole genome of making a type 1 error. All right? Does that make sense? Okay. So each individual point has to be extremely stringent to have a P05 for the whole genome. So that kind of stringency is tough to hit. And also, as I intimated way back in talking about all this, what this really is a fishing expedition for genes that might be related to your phenotype. Okay? So in order to give us a little slack here, and since it is a fishing expedition in reality, we're also going to look at what's called suggested individual. Okay? And so that's going to be a lot less stringent. For each individual point, that's only a P equal to 0.008. But that, across the whole genome, gives us an alpha error rate of 0.63. All right? That's pretty hefty. You know, that means that's a, yes?
How many iterations does this happen? Because I guess they're both the same number of iterations to go from 0.006 to become an alpha of
0.05, right. When I did this, I think there were about 743 markers, and I think we're up to about 4,000 right now. So now it's even a lot more stringent for both of these. Okay. By the way, I should go back and oops, sorry. Let's go the other way. So when we get our QTL maps in lab this week, you'll get one of the things that we'll get is a graph like this. This again is the likelihood ratio statistic as a function of all these markers on all the different chromosomes. And this blue line up here, this is the significance line. So if anything gets up there or better, that's significant. Okay. And this green line is the suggested. And I'll tell you right now, you guys didn't get anything that's significant, but you did get some peaks that are suggested. So, but for me, that's good enough. All right. And I'll explain why. Okay, so as we've already said, lots of error variants will give us false negative, type 2 errors when we do QTL analysis. So if we have lots of error variants, what will that do to the number of peaks that we get? Will we get fewer or more than we should? Fewer, right, okay. So that's a problem. If we have a lot of error variance in our data, we're going to get fewer peaks than we ought to get. All right? And hang on to that idea. Okay. Let's talk about the statistic itself. And I'm going to trot out the formulas here, and then we'll again go back and think about what error would do to this. Basically, the likelihood ratio statistic is a ratio. Any of you study analysis of variance when you took statistics? Any of you study analysis of variance when you took statistics? Whew, okay. All right. So basically, this is a ratio of, again, breaking variance down into two ways, essentially. All right? And the numerator here, what we're going to do is look at the variance with everybody. Doesn't matter what if they have the B or the D marker at that point. Okay? What we're going to do in the bottom is we're going to break that variance up. Okay? And the way we're going to do it is we'll look at the variability of everybody who had the B marker away from their mean, and then everybody who had the D marker away from their mean. All right? And then add it up. Okay? Anybody need? I'll give you a little more time to copy that down. Okay, let's just think about this. I've made up some data here, and again, what we're going to do is we're going to have the phenotypic measurement, a residual, in our case, and what the statistic does is take everyone who's got, in our case, the B marker versus the D marker. It says, okay, everybody's got the B marker, you're in this pile. Everybody's got the D marker at this point, you're in this pile. Now, let's go back and look at our numerator, and that says, okay, we're just going to look at the variability of everybody. All right? So what's going to be in the numerator then is here is the average of everybody, regardless if they were B's or D's. We're going to figure out the distance from there and square it, add it all up. Okay? And now let's go back and look at the denominator. And so what we're going to do here is we'll take each point that's a B and subtract it from the mean of the B's. All right? So we'll take each, here's the mean of the B's right there. We'll take each point, subtract it from the mean of the B's, square that distance, add it up. Check. Similarly, we're going to take each D from the mean of the D's, square that distance. So here's the mean of the D's. Take all these distances, square them, add them up. All right. So, you guys, if we did that, we took the variance of everybody, regardless of their B's and D's, or the variance from the B's and the variance from the D's, what's that ratio going to be pretty much? 1. Good answer. All right. And so let's go back to ye old formula here. And so this is the log of this ratio. The log of 1 is 0. Good. You got your money's worth when you took math. All right. Okay, so that's when the likelihood ratio statistic is low. All right, when the data are pretty much all spread out like that, B's, D's. Okay. Let's look at what it would look like if the likelihood ratio statistic is high. Okay, here we're going to do the same number. We'll take everybody and find the variance of each point away from the mean of everybody. Okay? And then we'll find, we need to separate get the variance just for the B's, each of these points away from the mean of B's, and the variance just for the D's, each of these points away from the mean of D's. Okay? In this case, what is this ratio going to be? Is it going to be close to 1? No. Will it be more than 1? Yeah, it's going to be more than 1. All right. And so that's what it looks like when the likelihood ratio statistic is high. We have the B's and D's will cluster together and be widely separated in the phenotype or the residual, whichever our measure actually is. Okay, so that's what the data look like when the likelihood ratio statistic is high. All right. Now, think about it. Think about this. Let's say this is our reality. This is what's true, but we got a lot of error variance because people drew things badly, and so now we're going to randomly add or subtract things to each of these points. Are these data going to still look like this? No. These data are going to turn around and look like this, right? If you start randomly adding and subtracting numbers. So that would be an example of what kind of error? 2. Type 2 error, yeah. All right, so a false negative. All right, so if there's a lot of error in our data, we're going to get more type 2 errors. All right, let's, in conclusion, talk about why we may not replicate Williams et al., the paper that's in your reader. All right? First of all, they used weight. They had, if you read their paper, they had a technician with the razor blade pop off the olfactory bulbs, chop them off, plop them on a scale. All right. So, one technician, one razor blade, one scale. Do you think that relative to you guys, who are all of you are drawing it, lovely, unique people, all doing it in your own way, do you think you're going to have more variability, or she's going to have more variability with her razor blade? We're going to have more variability. What will that mean for us relative to them? More or fewer type 2 errors? More. So we may get fewer peaks than Williams et al. No, I put up the answer there. Jeez. Okay, yeah. We may, so they probably had less error and fewer false negatives, or they made fewer type 2 errors, so we'll probably make relatively more. What they fess up to is when they looked at those brains, said, oh, we're going to recut through the anterior olfactory nucleus in an irregular fashion. Sometimes it's with it, sometimes it's not, okay? And what I did when we defined what is and is not olfactory bulb, we always included the anterior olfactory nucleus. So we may get slightly different results than they got, okay? All right, in this lecture, we learned about simple and multiple regression, how they can be used for controlling extraneous variables, how error is being controlled in your experiment, and a couple of reasons why you may not replicate Williams et al. All right, and I thank you for your kind attention. I'll see you in lab. Oh, we got to announce office hours, right? Okay, so the next assignment that is due. So this week, we're going to get you through everything. You already have the questions for the write-up on the course website. And so a week from your lab. So if you're a Tuesday person, a week from Tuesday, you have an assignment due. Wednesday person, you'll have an assignment due. Just answer the questions, no methods, results, all that. Please, just the number and the answer. And so to get you ready for this experience, can we turn on the lights, somebody, please? To get you ready for this experience, we'll have some office hours. And so on this Thursday, we got Rick, all the office hours are down in A456 Franz, the good old lab. Rick's going 11 to 1, and then Rick's back for a reprise on Monday. He's going 12 to 2, and I'm going to pick up 2 to 4. All right, so that's office hours. See you in lab.